You turn on your shower in the morning, or, you walk past one of those massive decorative fountains in a city plaza.
Oh, right.
Or maybe you're just like sitting in an air-conditioned office building. You probably aren't thinking about microscopic warfare,
Oh,
right?
definitely not. You just expect the water to be clean.
Exactly. You expect the water moving through all that infrastructure to be treated, clean, perfectly safe. But the reality of our built environment is actually, well, quite a bit darker.
Yeah, it really is.
Because the very infrastructure we rely on, our plumbing, our cooling towers, hot tubs, it might actually be harboring a microscopic hitchhiker just, waiting to be inhaled.
It is a profoundly unsettling thought. And when you look at the microbiology, you realize we've basically accidentally built the perfect artificial ecosystems for this ancient organism.
Right.
Like, we took a bacteria that was perfectly content living in pond scum, and we essentially built, a multi-million-dollar incubators for it.
Which is wild. So welcome to today's deep dive. We are unpacking the fascinating and honestly terrifying biology of Legionella, which is this water-loving pathogen responsible for Legionnaire's disease.
Yes.
We're going to get into how this bacteria hijacks human cells, how it totally evades our best water treatment systems, and how our modern conveniences have inadvertently armed an organism that never even meant to attack us in the first place.
It's quite the story.
It really is.
Yeah.
But, to understand what's happening in our plumbing today, we have to look back at how we even realized this pathogen existed. Because for a very long time, this was just an invisible killer.
It was. And the origin story of this pathogen, it really is like a medical thriller.
Oh, totally.
You have to put yourself in the mindset of July 1976, Philadelphia. There's this massive gathering at the Bellevue Stratford Hotel.
Right, the American Legion Convention.
Exactly. Roughly 4,000 members of the American Legion are there for the state's bicentennial convention. It's a huge celebration. But within days of the convention wrapping up, attendees start falling severely ill.
Like high fevers,
Yeah.
terrible coughs.
Yeah, and deep, very severe pneumonia.
And reading through the historical accounts, the escalation was just terrifying. Within a few weeks, 182 people who attended that convention were sick, and 29 had died.
Which is just a massive fatality rate for something spreading that fast.
Right. The media immediately dubbed it Legionnaire's disease, but nobody actually knew what was causing it. Public health officials were flying totally blind.
Oh, completely. People were speculating it was a localized outbreak of swine flu.
I read that others thought it might be like toxic chemical exposure, like nickel carbonyl poisoning from the environment.
Yes, there were even theories floated about intentional sabotage. I mean, the detective work to solve it took months and months of agonizing dead ends.
Wow.
It fell primarily to a microbiologist at the CDC named Joseph McDade, who was working with his colleague Charles Shepard.
Really?
McDade was looking at tissue samples from guinea pigs that had been inoculated with the patient's lung tissue, and he actually saw these tiny rod-shaped structures early on in the investigation.
Wait, he saw them early on?
He did, but he initially dismissed them as random environmental contaminants.
But why dismiss them? If you're hunting for a killer bug and you see a bug, shouldn't that be, like, the prime suspect?
Well, you would think so, but standard microbiology kind of betrayed him here.
How so?
When McDade tried to apply standard gram stains, the conventional chemical dyes we use to categorize and make bacteria visible under a microscope, these rods just didn't hold the color.
Oh, weird.
Yeah, they remained essentially invisible. And furthermore, when he tried to grow the pathogen in standard hospital culture media, the Petri dishes stayed completely empty.
So it's just a ghost.
A microbiological ghost, yeah. It completely defied the standard operating procedures of a 1970s pathology lab.
So it's not that the bacteria was too small, it's that its chemical makeup was actively resisting the tools they used to find it.
Right.
I imagine it's like a forensics team dusting for fingerprints. But the suspect happens to excrete an oil that specifically dissolves the fingerprint powder. The evidence is there, but your tools are erasing it.
That's a great analogy. The problem really comes down to the bacteria's unique cellular structure. Legionella has a highly lipid-rich outer membrane, and it spends a lot of its time hiding inside other cells.
Okay, so standard dyes just wash right off.
Exactly. It wasn't until the Christmas holidays that year that McDade, acting on a hunch, decided to go back to those failed slides.
Good for him.
Right. He used something called a Gimenez stain. Now, this is a very specific, aggressive dye, normally reserved for a totally different class of intracellular bacteria called Rickettsia.
Oh, so it penetrates differently.
Yes, and suddenly, against the contrasting background, there it was. The organism was clearly visible, swarming in the tissue.
Okay, so the Gimenez stain finally lights it up. But seeing it is only half the battle, right? If it wouldn't grow in standard Petri dishes, how did they finally manage to culture it to actually prove it was the culprit?
Well, they discover this bacterium is incredibly fastidious.
Fastidious?
Yeah, it's a very picky eater. It lacks certain metabolic pathways, so it absolutely requires high ambient levels of cysteine, which is an amino acid, and iron salts to grow.
And without those?
Without those freely available in the environment, it just won't multiply at all. This realization led to the creation of a very specific dark nutrient jelly called buffered charcoal yeast extract agar, or BCYE.
Charcoal. That sounds intense.
It is. The charcoal actually helps absorb toxic oxygen radicals that would otherwise kill the bacteria, while the yeast extract provides the cysteine and iron. Decades later, BCYE is still the gold standard for culturing Legionella.
Wow. And the real twist in the narrative is what happened once they finally had that BCYE agar.
Oh, absolutely.
Like they grew this new pathogen, figured out its profile, and then started testing blood serum from older unexplained pneumonia outbreaks.
Yes.
And they realized they hadn't discovered a new bug at all. It was responsible for an outbreak in 1968 in Pontiac, Michigan, among health department workers.
Which is actually why the milder version of the illness is called Pontiac fever.
Right. And an even earlier cluster in Washington, D.C. in 1965.
It had been circulating among humans for a long time, just completely undetected, which naturally raises the question, where had it been hiding before it found its way into those buildings?
Right. Where did it come from?
The answer to that reveals the central defining characteristic of Legionella. It was hiding where it has always lived, in natural freshwater ecosystems.
Like lakes and streams.
Lakes, streams, soil moisture. But it wasn't just floating around aimlessly in the water, it was residing inside other organisms.
Ah, right. So Legionella is fundamentally an intracellular parasite of amoebae. We're talking about single-celled organisms like acanthamoeba that live in pond scum.
Exactly.
These amoebas roam around grazing, swallowing up environmental bacteria for food. But Legionella evolved a way to ensure that when it gets swallowed, it doesn't become lunch.
It executes this brilliant and just aggressive defense mechanism. When an amoeba engulfs Legionella, it traps the bacteria in a digestive compartment called a phagosome.
Okay, phagosome.
Normally, the amoeba would fuse that compartment with a lysosome, which is a sac full of digestive acids, and just dissolve the bacteria.
Like a stomach.
Right, exactly like a tiny stomach. But Legionella uses a microscopic structure called a type 5e secretion system, specifically known as the dodexum system.
Dodexum. Got it.
Think of it as a complex molecular syringe spanning the bacteria's cell envelope. The moment it gets swallowed, Legionella uses the syringe to inject over 300 different effector proteins directly into the amoeba's internal cytoplasm.
Wait, 300 proteins injected instantly. That is a massive payload. What are all those proteins actually doing? Are they just neutralizing the acid?
It goes much deeper than just neutralizing acid. Those proteins actively hack the amoeba's internal trafficking system.
They hack it.
Yeah, they intercept transport vesicles that are normally moving from the amoeba's endoplasmic reticulum. The bacteria forces these stolen vesicles to wrap around its own compartment.
Oh, wow.
It basically builds a camouflage tent out of the amoeba's own cellular material.
Because it's coated in the amoeba's own proteins, the lysosome doesn't recognize the compartment as containing foreign food anymore.
Exactly. The fusion never happens. Legionella then sets up a private, protected replication chamber, multiplies wildly until it exhausts the host's nutrients, and eventually bursts out, killing the amoeba.
So it evolved this incredibly complex 300-protein molecular heist just to survive in pond scum. But if its entire evolutionary history is an arms race against amoebae, why does it absolutely destroy human lungs?
Because of a tragic evolutionary coincidence.
Oh, no.
Yeah, when a human inhales a microscopic water droplet containing Legionella, the bacteria travels deep into the lungs, into the alveoli. Your immune system recognizes a foreign invader and sends out its own defenders.
The alveolar macrophages.
Right, the alveolar macrophages, to engulf and destroy the bacteria.
And human macrophages operate the same way amoebae do, right? They swallow the target and try to fuse it with an acid sac.
Exactly. The cellular machinery that our macrophages use to eat pathogens is highly conserved across evolutionary history. It is chemically and structurally very similar to the machinery that ancient amoeba use.
Oh, I see where this is going.
Yeah, so when the human immune cell swallows Legionella, the bacteria just executes its standard programming. It doesn't know it's inside a human. It thinks it's inside an amoeba.
Wow.
It deploys the molecular syringe, injects the effector proteins, halts the digestive fusion, wraps itself in our endoplasmic reticulum vesicles, and starts multiplying inside your lung cells.
That is wild. Human lungs are basically just an accidental training ground for a bug that evolved to fight pond scum.
Exactly.
We aren't even the main character in this bacteria story. We're just collateral damage in an amoeba war.
We are what biologists call a dead-end host. The bacteria gains zero evolutionary advantage from infecting us.
Because it kills us.
Right. Right. Because it causes severe pneumonia, the host often becomes incapacitated or dies, and crucially, humans do not pass the bacteria on to other humans.
That's a great point.
It's purely an accident of biology that our immune cells look like its natural prey.
I want to highlight that point because it clears up a massive misconception. If I'm standing in an elevator next to someone who is coughing their lungs out with severe Legionnaire's disease, I am not in danger of catching it from them.
You are safe from Legionella in that scenario. There is essentially zero person-to-person transmission.
Okay, good to know.
You cannot catch it from someone coughing on you. You can only get it by inhaling aerosolized environmental water that contains the bacteria.
But if you do inhale that water, what actually happens to your body? Because like we mentioned earlier, this one organism causes two completely distinct diseases.
Yes, it does.
There's Legionnaire's disease, which can put you on a ventilator, and then there's Pontiac fever.
The clinical presentation is a fascinating medical puzzle. Pontiac fever is a mild, self-limiting, flu-like illness. You get a fever, muscle aches, fatigue, but there is absolutely no pneumonia.
So no lung issues.
Right. Your lungs remain clear, and it usually resolves on its own without any targeted medical treatment in a few days. Legionnaire's disease, conversely, is a severe, deep-lung, atypical pneumonia with a notable mortality rate if left untreated.
So why does one person end up in the ICU with failing lungs and another person just feels crummy for a few days? Is it like a different mutated strain of the bug?
Nope, it is the exact same organism. The difference comes down to the dynamics of the exposure and the host.
Meaning?
It's a combination of the inoculum size, how much bacteria you actually inhaled, and the robustness of your immune system. Most healthy people exposed to Legionella either get Pontiac fever or clear the bacteria without ever showing symptoms.
Oh, wow. So who gets the severe version?
The severe pneumonia usually strikes individuals with specific vulnerabilities. So people over 50, current or former smokers whose lung tissues are already compromised, or those with underlying conditions like COPD or weakened immune systems. Okay,
let's talk about treating those severe cases. Because if a patient comes into the ER with raging pneumonia, the standard protocol is to hit them with broad-spectrum antibiotics like a beta-lactam, your penicillins or cephalosporins.
That's the standard, yes.
But those standard drugs just bounce right off Legionella, right?
They do. And understanding why requires looking at antibiotic pharmacokinetics.
That's a big word.
Basically how the drug moves in the body. Beta-lactam antibiotics work by interfering with the synthesis of peptidoglycan, essentially destroying the bacterial cell wall, but they are largely confined to the extracellular space.
Outside the cells.
Right. They float around in the blood and the interstitial fluid.
And Legionella isn't out in the blood. It's hiding inside the human macrophage, wrapped in its stolen camouflage tent.
Exactly. The standard penicillin just floats on by, completely unable to reach the pathogen.
So how do doctors even fight back?
To treat Legionnaire's disease, physicians must use specific classes of antibiotics, primarily fluoroquinolones like ciprofloxacin or levofloxacin or macrolides like azithromycin.
What makes those different?
These drugs have chemical properties that allow them to easily cross human cell membranes. They penetrate the macrophage, cross into the Legionella-containing vacuole, and disrupt either bacterial DNA replication or protein synthesis from the inside.
So you need an intracellular drug for an intracellular bug. That makes perfect sense. We know how it fights us, and we know how to fight it medically.
Yes.
But the truly unsettling part is where we are inhaling it from. I mean, we aren't out sniffing pond scum. We are catching this from our own modern infrastructure.
The built environment is the ultimate reservoir. Legionella thrives in the complex water systems humans engineer.
Like what?
Massive cooling towers on top of skyscrapers, the intricate premise plumbing systems in hospitals, hotels, cruise ships, hot tubs, large decorative fountains.
Now, here is where I think a lot of people get confused. City water is heavily treated. Municipalities pump chlorine or chloramine into the water supply, specifically to eradicate pathogens.
Yes, they do.
How is this organism surviving a chemically treated water grid?
Well, this exposes a critical gap in our infrastructure management. Municipal water utilities generally do an excellent job of delivering treated, disinfected water to the front door of a building.
Okay.
But the moment that water crosses the water meter and enters premise plumbing, the vast network of pipes, heaters, mixing valves, and tanks inside a massive facility, the flow dynamics change, and the responsibility shifts entirely to the building owners.
Oh, I see.
The chlorine residual from the city often dissipates by the time it travels deep into a complex building.
And even if there is some chlorine left, people assume chlorinated water equals sterile water. But Legionella has adapted to survive chemical attacks, primarily by utilizing biofilms, right?
Biofilms are a massive problem in plumbing. In large buildings, there are inevitably areas of low flow or dead legs.
Dead legs.
Yeah, pipes that were capped off during a renovation, for example, where water just sits stagnant. In these areas, various microbes excrete a sticky, complex matrix of extracellular polymeric substances, or EPS.
Oh, gross.
Yeah, they build these slimy communities on the inside walls of the pipes. Legionella shelters deep inside these biofilms. The EPS matrix acts as a chemical shield.
So the chlorine doesn't even reach them.
Right. As chlorine flowed past, it reacts with the outer layers of the biofilm and gets consumed before it can penetrate deep enough to reach the Legionella.
So the biofilm is a physical and chemical bunker. And on top of that, you still have the amoebae in those pipes. So the bacteria is hiding inside a bulletproof vest, inside an The manager can flush the system with chlorine, but the bacteria just rides it out.
That layered defense is exactly why it is so notoriously difficult to eradicate once it colonizes a building's plumbing. And this biological reality collided with our infrastructure in a very dramatic way during the early days of the COVID-19 pandemic.
Because the lockdowns created the ultimate stagnation event. Offices, schools, gyms, hotels, they were abandoned overnight.
Millions of miles of premise plumbing suddenly stopped flowing. The continuous movement of water and the regular influx of fresh municipal disinfectants just, ceased.
Right.
Water sat in these massive pipes at room temperature, undisturbed, for weeks and months. The chlorine residuals plummeted to zero. It was a perfect global incubation event for Legionella.
Wow. I hadn't even thought about that.
Infection control experts and environmental engineers were terrified. Before those commercial buildings could safely reopen, they required intense flushing protocols, sometimes superheating the water or applying massive shock doses of chlorine.
Just to break through the biofilms that had established themselves during the lockdown.
Exactly.
It's a stark reminder of how fragile our control over our own infrastructure really is. And when that control slips, the consequences are deadly. We don't have to look far back in history to see this. The 2015 South Bronx outbreak is a perfect example of how an infrastructure failure translates directly into a public health crisis.
Yes, the South Bronx outbreak is a landmark case study in environmental epidemiology. In the summer of 2015, a severe cluster of Legionnaires disease cases rapidly emerged in New York City.
And investigators had to figure out what all these disparate people had in common.
Right. They eventually traced the aerosol as bacteria back to a contaminated cooling tower sitting on top of a hotel.
For anyone who doesn't know how a cooling tower works, it's a crucial part of a large building's HVAC system. They use the evaporation of water to remove heat from the building.
Yes.
But in doing so, they use massive fans that exhaust air out of the top.
Yeah.
And that exhaust contains drift, microscopic water droplets.
Yeah.
So this cooling tower was essentially functioning as a giant contaminated aerosol generator, just blowing an invisible mist of Legionella down into the streets.
People just walking by on the sidewalk, waiting for a bus or walking to work were inhaling this microscopic drift. That single outbreak sickened over 100 people and killed more than a dozen.
That is tragic.
The public outcry was immense, and it led to immediate political action. It forced New York City, and subsequently the state, to pass the very first mandatory cooling tower registration, testing, and inspection laws in the country.
Wait, before 2015, they didn't even know where they all were.
Before 2015, the city didn't even know where all the cooling towers were located. The South Bronx outbreak is a rare, clear-cut example of a microbiological event forcing the rapid wholesale creation of new municipal engineering laws.
It's staggering that it took a deadly outbreak just to mandate a registry of where these machines were located. But New York isn't the only major crisis tied to this pathogen.
No, it's not.
There's another event that everyone has heard of, though mostly for a different reason, and that's the Flint, Michigan water crisis.
The Flint water crisis, which unfolded heavily between 2014 and 2015, is internationally recognized as a catastrophic lead contamination crisis. The city switched its municipal water source from the Detroit system to the Flint River as a cost-saving measure, but they failed to apply adequate corrosion control treatments.
Right.
The highly corrosive river water stripped the protective scale from the city's aging pipes, leaching toxic levels of lead into the drinking water.
But parallel to the lead crisis, there was a massive lethal spike in Legionnaires' disease cases in Genesee County.
Right.
We're talking about dozens of severe pneumonia cases and multiple deaths happening at the exact same time the water chemistry changed.
The timing was undeniable, and the proposed mechanism linking the two is fascinating from a chemical standpoint.
Okay, walk us through it.
Well, many epidemiologists argue that the lack of corrosion control didn't just leach lead. It fundamentally altered the biological stability of the water. The highly corrosive water stripped away the chlorine residual much faster than normal. Furthermore, as the iron pipes corroded, they released massive amounts of iron into the water supply.
Oh. And as we established with Joseph McDade's BCYE agar, Legionella absolutely requires iron to thrive.
It was a perfect storm. The hypothesis is that the municipal water switch effectively stripped the premise plumbing of its chemical disinfectants while simultaneously fertilizing it with a massive influx of iron, allowing Legionella to proliferate wildly in hospitals and homes.
Now, I do want to make sure we represent the full scope of the research here.
Absolutely.
It is important to note that the epidemiological community still debates the specific attribution of every case, right?
Yes, very true. Some researchers argue that severe, localized plumbing failures within a major regional hospital were the primary driver of the largest cluster, complicating the data.
Right. There's an ongoing scientific debate about how much of the outbreak was due to the municipal switch versus specific building management failures. But I mean, either way, it illustrates how deeply interconnected water chemistry and microbiology are.
Without a doubt.
You change a municipal water source to save a few bucks, you alter the pH or the corrosion control, and suddenly you cause downstream effects that strip chlorine and feed a deadly pathogen in a hospital shower miles away. You can't just tweak one variable in a complex system without risking cascading failures.
Flint is a tragic masterclass in the unintended consequences of infrastructure management. And looking forward, the challenges of managing Legionella in our built environment are only becoming more complex.
Why is that?
It all comes down to temperature. Legionella has a specific thermal sweet spot. It survives in cold water, but it multiplies rapidly in water that is between 25 and 45 degrees Celsius. That's roughly 77 to 113 degrees Fahrenheit.
Which is essentially the temperature of warm tap water. It's exactly the temperature we want our showers and our hot tubs to be.
It is. And consider the modern push for green building design. Hospitals, hotels, and massive residential complexes are constantly trying to balance safety with energy efficiency.
Because heating thousands of gallons of water takes an immense amount of energy.
Right. So to lower costs and reduce carbon footprints, many facilities lower the baseline temperature of their massive water heaters. Furthermore, they use complex mixing valves to ensure water delivered to a patient's room isn't hot enough to cause scalding.
Wait, but by lowering the baseline temperature in those massive premise plumbing systems to save energy, they are dropping the water straight into that 25 to 45 degree thermal sweet spot.
They are inadvertently engineering the optimal environment for Legionella to multiply.
Wow.
The very systems designed to make our buildings environmentally friendly and comfortable are acting as massive bacterial incubators. Add to that the broader macro context of climate change, which is slowly warming ambient fresh water sources, lakes, rivers, and reservoirs globally.
So warmer source water means a higher baseline population of Legionella and its amoeba hosts entering the municipal treatment plants in the first place.
Exactly. Public health officials are watching these trends very closely because the convergence of warmer source water and energy-efficient building temperatures is a significant vulnerability.
It is a phenomenal reality check. We think we've conquered nature with our concrete and our pipes, but we've really just built a new, highly optimized jungle for an ancient predator.
That's a great way to put it.
Thank you for walking us through this incredible biological detective story and unpacking the mechanics of how this pathogen actually operates.
It's a vital topic. Understanding how these organisms exploit our environment is the first step in engineering better defenses.
And to you listening, thank you for joining us on this deep dive. As we wrap up, I want you to think back to that shower you took this morning and leave you with this one final, unsettling question to mull over.
Oh boy.
As we push to build greener, more energy-efficient buildings by lowering the temperatures in our massive water networks, are we inadvertently engineering the perfect incubators for Legionella? In our vital fight against one existential threat, climate change, how do we ensure we aren't accidentally arming an ancient microbiological enemy hiding right inside our own showerheads?